Hello, everybody. It's Mrs. Pound, and I'm back with more from Chapter 1 from BJU Biology, 4th edition, about the science of life and the God of life. And in Section 1B, we're going to talk about the scientific method. So our objectives for today are going to be to list and describe the significant steps of the scientific method, recognize the limitations of science, describe a Christian philosophy of science, and explain the difference between applied and pure science. So let's start with the scientific method and using it. Now, one thing you have to realize about the scientific method is a lot of times the way we teach it, it sounds like you start with step one and you end with step Z. Okay, start with step A, end with step Z, and it's linear. That is not the truth. Sometimes we jump around on the steps. Um, Sometimes we eliminate steps, sometimes we have to go back and repeat steps, but it does give kind of an outline to give us some guidance when we're doing investigations. So the scientific method is a logical method of problem solving that involves observing and reaching a conclusion. A lot of times, a scientific investigation starts with research. This is an investigation into a topic often carried on by reading, inquiry, or scientific observation. A lot of investigations do start out in the library, reading about what other people have done so that we can build upon knowledge and not recreate the wheel every time. Many times a hypothesis is formed. This is an educated guess about the solution to a problem and helps define and direct an experiment. So we don't always have a hypothesis, but they are helpful because it gives direction for the experiment. And a scientist's hypothesis may not always be true. Okay, their best educated guess may not be correct. So it's okay not to be correct when forming a hypothesis. A hypothesis must be a simple, testable statement, and it predicts the results of a research study. So it is a prediction, and not all our predictions are correct. Now, in order to determine if the hypothesis is correct or not, we have to collect data. The recorded information from an experiment or a survey. So we need information, and set an experiment or a survey, we do have a couple of possibilities. Now, we could conduct a survey. Let's say we want to know about the bird population in an area. Perhaps maybe rattlesnakes have been reintroduced to an area, and we know that rattlesnakes eat organisms that prey upon birds. And so we're going to hypothesize that the bird population will actually increase with the introduction of rattlesnakes. And so there's no experiment really here. We're just observing. And so we're just observing. And so we will conduct a scientific survey in order to see what is going on. We could also perform a controlled experiment at times. And a controlled experiment is an experiment testing two identical groups for a single variable. So we see bacteria in the background, maybe they've been treated differently. And so we're seeing which grows the more bacteria. In a controlled experiment, we have what is called the independent variable. This is the variable in a controlled experiment that is manipulated by the researcher. It's also called the experimental variable, excuse me. So here we have a syringe that's being inserted into the tomato with, I don't know what it is, maybe some kind of chemical. And that is the independent variable because we're treating it with something. This is the difference between the control group and the experimental group, and it's to reduce the possibility of error. A scientific experiment should not have more than one variable. So we're going to only have one independent variable because if we have two, then we don't know which one of the two is actually causing the change that we want to measure. We also have a control group. This is a group in an experiment that is not exposed to the experimental variable. So these tomatoes here are not being injected. They are the control group. The experimental group is injected with the independent variable, so this is the group in an experiment that is exposed to the experimental or independent variable. And then there's a dependent variable. This is the variable in a controlled experiment that shows the effect of the treatment. This is what we're measuring. So after we treat it with the independent variable, we're going to cut open the control group tomatoes. We're going to cut open the experimental group tomatoes and see if there's a difference between them, a measurable difference. Maybe we've injected something into the tomatoes that we think is going to affect taste. So we'll taste the tomatoes, or maybe it's how long they last. So we're looking to see what happens. Now, if the results of a scientific experiment are accurate and reliable, they are considered valid. Let's take a look at spontaneous generation and the scientific method. We're going to take a look at some experiments that were performed to try to prove and disprove spontaneous generation, which remember is where non-living things arise, or excuse me, where living things arise from non-living things. Van Helmont, he performed an experiment where he believed the sweat of a sweaty shirt supplied the active principle needed to cause mice to generate in a box. And so in his experiment, he did not actually have enough controls. He put a sweaty shirt in a box and mice appeared. And so he said that the sweaty shirt produced the mice. He had no other controls. He didn't like put the box inside of a box that the mice couldn't get into. It was just like more a paper box that they really could chew through and things like that. So he didn't have enough controls to make sure that outside mice were not getting inside. Spallanzani demonstrated that the active principle of an infusion is not destroyed by boiling. So it was the active principle that was believed that produced the organisms. And what he did was he made an infusion, okay, using wheat and boiling it. And after boiling it, he let it sit. And he discovered that things grew in it even after boiling. Again, he didn't have enough controls because he left it open to the air. Okay. But Louis Pasteur came along, a Frenchman, and he developed this swan-necked flask. And with this flask, it had so many controls on it that the results disproved spontaneous generation to the satisfaction of most scientists of the time. What the swan neck did is it allowed him to boil the infusion and kill the microorganisms that we now know live in there. But after it boiled it, the microorganisms that were in the air outside could not make it through the neck and work against gravity and go back into the infusion. Um, in fact, his infusion did not spoil until he broke the neck, and then those organisms could get in there to grow. So he disproved spontaneous generation. Now, one thing that we need to know is that spontaneous generation must be included in the theory of evolution because it requires non-living elements to combine to form complex molecules, proteins, nucleic acids, in order to form a living cell at least once. Okay? So, if you believe in evolution, you believe that spontaneous generation happened at least once because uh, you would have to say that something non-living did form something living. So let's take a look at some of the limits of science. First, to understand the limits, we have to know what a phenomenon is. And a phenomenon is something that can be observed or measured. It may be an object, property, or process. So a natural phenomenon might be something like a tornado that we see in the background. Now, science cannot be used to explain a phenomenon, only describe it. So, science only describes this. It does not totally explain it. It might describe that there's differences in air pressure. It might describe what it looks like. It can describe the conditions that cause it, but it cannot completely explain it. Also, in science, there tend to be biases. This is a preference or inclination in thinking, especially a belief that causes one to be partial. So, a person's beliefs can cause bias, maybe whether they believe in biblical principles or they do not. Also, you see that dollar bill there? money tends to influence people and bias them. So there is bias in science. So interest outside can leak in. Man is prone toward bias in his interpretation of facts. Also, workability. Workability is a characteristic of scientific knowledge that allows its practical application. So we're looking for workable explanations, and it can be workable or not depending upon your bias at times. One of the primary goals of scientific work is to find information that is in fact workable, so that is useful for us, that explains things, that can predict them. Also, man cannot be totally depend on his observations. Sometimes our observations are flawed because our techniques are not adequate. Our equipment isn't as advanced as it should be. Um, that type of thing. For instance, with the spontaneous generation, they didn't have microscopes to see the microorganisms. I also saw a really good cartoon on this, where maybe we're just not observing a big enough picture because there the cartoon shows two men, one looking over a wall, one looking through a hole in the wall. And the man looking through a hole says, "Look, it's raining out." The man looking over the wall says, "No, it's not. There's a man with a hose." Okay, so depending on how much you can observe of a situation, um, could lead you to not have dependable results. Also, no scientific experiment can be completely controlled, especially with biology. We just cannot control all the variables. So sometimes there's an extra variable that gets in there that changes things that we don't realize. Also, scientific investigation cannot prove or disprove a universal statement. And so, what's a universal statement? These are words such as all, always, no, or never. These are also those words we tell you to be careful of on true false questions or even on tests, these universal statements. A lot of times they are false. Why is that? Because it's impossible to observe every situation or area of the world to determine the absoluteness of the statement of humans. So we could say all or always, but we cannot observe everything, so we don't necessarily know. So, what does that mean for science and the Christian? Okay, pure science is the knowledge obtained through scientific activity. So, this is just we go out and observe and look around. This is what you did, what you do when you go outside and you're just making observations about the natural world. That is pure science and writing down your observations. Applied science is the use of pure science to solve practical problems. We do this all the time in medicine. We have developed medications and techniques to determine and help our health and to cure diseases. Now, there are also research methods. These are the use of scientific methods to obtain knowledge. So, this goes along with pure science. We're just using methods to obtain knowledge. So, we're just performing an experiment and um, to learn something. There are also technical methods. This is the use of prescribed techniques to gain knowledge about a specific case. So we have technical methods for analyzing blood to determine what's in the blood sample to determine an individual's health and well-being. Now, there is a scientific challenge to scripture. Science cannot be used to establish truth. Okay, science cannot be used to establish truth. It can tell us about the world around us, but it cannot tell us truth. Only the Bible can do that. So, a proper Christian attitude towards science should be one of worship. This is man's recognition of his significance and the resulting dependence on and praise to the Almighty God. Not that we're worshiping science, but since God is revealed in his creation, true science can enhance a Christian's worship of God. It can show us just how amazing God is because of the world that he made for us. So, our objectives were to list and describe the significant steps of the scientific method, Recognize the limitations of science, describe a Christian philosophy of science, and explain the difference between applied and pure science. So continue watching and answering those questions. And don't forget your 5 questions that you're to write in your margin for this video.